Assalamualaikum warahmatullahi wabarakatuh. In this video, we will discuss about soil, water, and airborne pathogen monitoring. A question: why we need to do monitoring uh, to the soil, water, or from the soil to the soil, water, and also in the air? Why? Yeah, let's really answer that question. So this diagram is, or this we call it, uh, general disease cycle. Yeah, general disease cycle. We start with attachment. Yeah, attachment is when the inoculum attach the surface of the host. Inoculum. What is inoculum? Inoculum is part of the pathogen, the infectious. For fungi, it can be a spore, hypha, sclerosia. For bacteria or virus, it can be the particle of the virus or individual of the bacteria or nematode. So it starts with attachment, and the inoculum attach the surface. Then the inoculum will be recognize the surface. Is that their host or not? If it's a stone or a wall or a door or a window or a glass, they will recognize that it is not my host. So they will not continue the process. They will hope they will can move by air or by wind or by water to the other area or surface. And hopefully they then attach in the host. Yeah. But if they recognize that it is their host, yeah, it is their host, so they will continue germination, and after that we will try to come in into the cell, or we call penetration, and after that there is interaction between plant and also the pathogen. We call it infection. Yeah. After that, the pathogen will grow in the plant and then make a lesion. Yeah, develop a lesion. Lesion, or what we also know as symptom. So produce symptom or symptom appear is almost late of this disease cycle. The disease cycle is almost finished. Yeah, almost finished. The symptom is stage is number. It's very late. And we know in previous meeting we learned about how to monitor disease by using incidence and also severity. And that monitoring is based on the symptom monitoring. So it's almost late because we cannot observe only if this cycle is almost done. Yeah, when the symptom is appear. So the idea is, can we monitor the pathogen before it? So let's go back. Yeah, go back into attachment. Yeah, we go back into attachment. We hope we can monitor the pathogen before it make a lesion. Yeah, we come back to the attachment. Before attachment, the inoculum need to move from one place to the other place. It's called dispersal. Dispersal. Yeah, and it can be moved by using water, air, and also soil. So if we can monitor the inoculum inside the water, air, and soil, so we can monitor it before it attach and make a symptom. Attach the plant and make a symptom. So that is the idea. The data will be indicator how much the inoculum in the area. It will be indicator how risk is this area for planting. So the next question is how we can do monitoring in the water, soil, in the air. Before it, yeah, when we taking a sample, make sure that we record all field information. Basic information like address, the phone number of the farmer, specimen number or code, yeah, the date, yeah, when you collect the sample. The time is also important, and what kind of crop there, yeah. And the information about the site, yeah, like a crop, size of planting, type of planting, type of plant, cropping practice, planting history. It will be help us, this data will be help us for diagnose the disease or for detection the pathogen, for detect the pathogen from the sample. Why? Because the disease sometimes have a unique interaction with the type of planting, yeah, with the type of plant, even with the cropping practice, yeah. The planting history will be help us to understand what kind of plant is planted before. It also will help us in the diet. Number three is about the disease. Symptom. What is the symptom? If you have phone or camera, take a picture or take a video there, yeah. Well, distribution of the disease percentage. How much is the incidence and the first appearance? It will be help us for diagnose the disease. Yeah. So please collect and record this data. Also, we will need farmer comment. What is the farmer comment for this situation? Maybe the farmer have idea about what kind of disease is it or the other. So please ask also the farmer comment. Okay, and after that, let's we collect the sample. There is several uh, point that we need to know about collecting sample. Number one is the pathogen population is very important. The pathogen population inside the water or in the air or in the water is very important. Yeah, because we need to detect it, we need to culture it after it. So the population is very important. Previously, when the sample still in the area, the population of pathogen is high. But after we collect it, yeah, the population of pathogen in the soil or water will decrease rapidly in the absence of living hosts. So if we take only the water or only the soil without the host, the population of pathogen will be decreased rapidly. And this will be make a difficulties for us to detect it later in the laboratory. So we have to think about it. Yeah. Second, pathogen quickly killed by freezing or heating. So if you transport it from a area or from the field to the laboratory, please think about how you bring it. Yeah, don't freeze it or heat it by incidents or by plan. So when you stop in the rest area, then you park your car in the parking station. Then the car had um, sun sunlight, then the temperature inside the car is high. This will be hit your sample. So please make sure that you sample in the good temperature. Yeah, to protect the population of the pathogen. If you take a sample for nematode, yeah, sample taken from 5 until 20 centimeter from the soil surface. Yeah. The sample was taken from the root zone of the plant showing symptom, but it's not dead. Yeah. So please do not take a sample from a dead plant or from the soil or water uh, surrounding the dead plant, because in that plant the pathogen is absent. Yeah, there is no pathogen anymore. Yeah, the pathogen is in a high population in the in the plant that still show the symptom. So if you put from healthy plant, there is no pathogen. But also if you put it from dead plant, there is no pathogen. So please take it from a symptomatic plant. Yeah, symptomatic plant. Take it from the root zone. Take a sample only when it's not too wet or dry. So soil, you cannot put the soil if the soil is very wet or dry. Yeah. Give a complete information, of course, we talked it before. Place the specimen in the plastic bag at the time of collection, so directly. Do not seal it in the plastic bag. Do not enclose moist paper towel. So you do not need to put a moisture condition. Just put it in the water in the plastic, like in the picture. Yeah. Do not wet the plant. Do not put water in the plant. Do not overstuff the plastic bag. So one plastic for one sample. Wrap the root or the soil ball securely, but not the leaf. Yeah. So this is the option. You can also take the sample that include the plant. So also the plant you put it in the laboratory to make sure the population of the pathogen is not decrease rapidly. Yeah, because they still have a living host there. Then send the specimen immediately after collecting them. So directly send to the laboratory. So if your laboratory is closed on the Saturday or Sunday, so don't take the sample in the evening. Better you take it in the Monday, so you can directly put it in the laboratory. Then what we can do in the laboratory? The challenge. Today we will, in this session, we will also become a laboratory person. Yeah? The challenge is physical difference between major pathogen groups. So to identify the pathogen, we do by using see the morphological difference or unique morphological unique that they have. But some of the pathogen there is no that do not have uh, unique characteristic from the morphological. So it's make it different. Number two is low concentration of pathogen. This is what we talk about. What we talked previously. The population of pathogen decrease rapidly because there is no living host. So it will be make it difficult here in the laboratory. Low concentration of pathogen. So the solution is we need to do enrichment. Enrichment is to increasing the population again by putting a nutrition inside the sample, so the population will be grow up. But all microorganism will be grow rapidly. The population will be grow rapidly. Not only the pathogen, but also the other microflora, the other microorganism will be also uh, increase rapidly because of this enrichment. Yeah? Because this enrichment methods sometimes is not specific for the pathogen. It's general for all microorganisms inside the sample. But this will be help us because the low concentration will we cannot do anything with low concentration except we use molecular detection. So that's why we need to do enrichment. The other challenge is inhibitor, yeah, from the sample. Especially if we use water, there is a polluted water that can kill our pathogen. So it also will be an inhibitor. Establish general protocol. So it's need a fixed protocol, standard protocol for do detection in the laboratory. But the problem is for one pathogen with other pathogen sometimes have different method of different protocol to conduct. So we have to do several tests for several pathogen suspected. Uh, After that, we can do the detection by using culture dependent method. Yeah, it's extensively used. Yeah, but it have low sensitivity. So low sensitivity because when we have a low concentration of pathogen, we cannot detect the pathogen. We cannot detect the pathogen. Yeah, we only can detect the pathogen when the concentration is very high. So it's why we call it low sensitivity. But sometimes it's also uh, good or better. This low sensitivity is better. Why? Because sometimes in the soil or in the water there is a lot of pathogen. Yeah, there is a lot of pathogen, and the major pathogen is the one that a lot in the soil or in the water. So I will repeat. In the soil or in the pathogen or in the water, sometimes there is more than one pathogen. Then only one that make our plant is sick from what? For the example, there are five pathogen. Only one of these five pathogen is the one that make our plant sick. So from these five, which one is the pathogen? So we can assume that the pathogen is the one that have the most population between these five. For the example, the pathogen A have the highest population. So it's probably is the causal agent that make our plant is sick because the population is very high. Yeah. So this is the we can assume like that. Nah, this culture-dependent method is low sensitivity, have low sensitivity. So it's mean they only can detect the one that have a high population. That is the A sample, the A pathogen. Because for pathogen B, C, D, E, the population is very low. This method cannot catch it because the population is very low. So the one that can catch, the one that can observe is only the A pathogen that have high population. So sometimes low sensitivity is better. Then after that, we will talk about molecular detection. It's very sensitive, but this make it difficult because all of pathogen can be detected, even they that have a small population. Yeah, we know that this small population doesn't mean anything because they will not make plant sick if the population is very low. But this molecular detection is very sensitive, so every pathogen there will be detected. Okay, it's still about culture dependent method. Number three is not all pathogen is culturable. So this method is we will culture the pathogen into the medium, but not all of plant pathogen can culture in the medium. So we cannot detect, we cannot process with this kind of method for pathogen that cannot culture in the medium. Before we culture it in the medium, we need to make a dilution first. Yeah? Sometimes for fungi can make dilution until 4. For actinomycet until 5, for bacteria until 7. Yeah. After make a dilution, then we pour. You can see in the slide, pour plate method. Yeah. First, we pour the sample. Yeah. The sample is the one that we dilute before. Yeah. We pour it in the petri dish, sterile petri dish, and after that, we pour a medium here there. Yeah. Then incubate it. And then after that, we will see the microorganism will grow in the medium, and then after that, we do identification. Now, there is several type of medium for fungi like OAES or VA juice or PDA. It's a common medium, PDA. Yeah. For actinomycetes, we can use water agar, SN glycerol. For bacteria, we can also use NA. There is three type of media. Number one is selective media. The selective media is generally select for growth. Yeah, so only the target microorganism that can grow in this selective media, because the selective media have can killing non-target organism. They have some toxic that can kill other microorganism, but not the target. So only the target that can grow in this medium. Yeah. Second is differential media. Differential media will not kill other organism, but for the target organism, the target will show unique colors, like the color will be very red or very yellow. It's indicate that is the target of our detection. But the other organism still can grow then. Yeah. But the the grow is just a normal, like a normal one. But for the target will be make visible change growth, yeah, like the color or the other. And type 3 media is general media. The general media is not selective. It's contain a lot of nutrition that need by microorganism. So almost all microorganism can grow in this media. But again, there is some pathogen that cannot be cultured in this media because they are parasite organism that can only grow in the living organism. So they can grow in this medium because this medium is not living organism. Now, this is a molecular method. Yeah. Uh, it's also can be used, and it's very sensitive. So if you have a sample and the population of the pathogen is decreased, is no matter, because this molecular method is very sensitive. So even there is only one inoculum, one spore in your sample, it can detect it. Yeah, and also very specific. Yeah, you can do detection specifically for a genus or for a species. Yeah, and it's also very fast than culture method. Molecular method is very fast to conduct. The other way is by baiting technique. It's a solution for a pathogen that cannot grow in the medium. Yeah, we can use a baiting technique. Baiting technique is method select for live active zoospore, which can swim toward infect the bait. Let me see. This is how we do a baiting. Yeah. So this is for water sample. So we put it in the receivable bag. Yeah. And we put the bait in the water, in the surface of water. We can use leaf, or also we can use a fruit like a pear fruit. And for several day incubation, if the leaf have a lesion or the fruit have a lesion, that is the pathogen that swim and then touch the bait and start to infect the bait. After that, we can do isolation. We can isolate the leaf or the fruit to grow the pathogen and to make sure that the pathogen is right. Yeah. Here, if we can collect the water, yeah. But if you want to monitor the pathogen in the river, you can put the bait or the leaf in the bag, and you float it in the river. Yeah, make make sure that you tied it in the trees or in the stone or in the wood. Yeah. So make sure you not lost it. Yeah. So you floating it in the river, and after several days, you take the baiting and see the baiting. If the baiting have several lesion, it means there is a pathogen in the river. The river. Um, or you can also take directly the fallen leaf. Yeah. Maybe in the near the river there is trees and the leaf is fallen and the leaf floating in the river for several days. Nah, you can take the leaf that show a lesion in the leaf. So we can also use it. This is for water, for soil. Yeah, this is for soil bathing. You can dip the potato directly uh, into the soil, or you put the soil in the box, and after that you pour a sterile water, yeah, and you leave it for a night to make the water clear. After that, you place the baiting in the surface of water or in the water. Yeah. You can use pure fruit, potato, leaf, and the other. It's also open for the other research to find what kind of leaf is effective for baiting some pathogen. Now we move to the airborne pathogen. The pathogen that move in the air common is spore from fungi. Yeah? So we can conduct monitor for spore. It can be conduct in the field, but it also important we conduct in the room. Yeah, if you have laboratory there or in your production house or in your house, you can make sure that your air inside the room, inside the laboratory, is clean from pathogen spore, yeah, or contaminated spore. Yeah, you can do monitoring. Just a simple monitoring, actually. Yeah, if you have um, general media, you put it in the petri dish. After that, you open the petri dish for 6 hours in the middle of the room. Make sure there is no people come there and see the medium. After that, you close the petri dish, then you seal it and incubate. In several days, you will see how many fungi or bacteria that grows. That is the bacteria that circulated in your air and then attach your PDA. Well, but this need a long time. It's maybe about 6 hours to conduct it. To make it fast, you can also use this kind of tools. Yeah, these tools have a vacuum that will flow the air from the up to the down, yeah, to the bottom. Yeah. So you can make the everything in your air directly attach your PDA fast. Yeah, because they have airflow that we make it fast. So you can do it just only 1 hour or a half hour, not for 6 hours. But in the field, you can use this type of trapping. Actually, it's similar. We open the medium in the open air and we wait for some hours and we see how many inoculum that attach our plate. Yeah. So make us can do interesting observation. Let's see when we do a sport trap at the 6, at the 10, at the 2 p.m., 6 p.m., 10 p.m., or 2 a.m., we can see that there is a difference of basidiospore that are couched in the trap. We can see that in the night, yeah, from 2 until 6 a.m., there is a lot of basidiospore that circulated in the air. So we have to be uh, so we can do some preventive, yeah, because in this time there is a lot of bacillus that circulated in the air. It's also we can um, compare it by the humidity condition or temperature condition. Sometimes it's affect the number of bacillus that move in the air. So this is observation in the field. Yeah, this is an observation in the field. Yeah, if you want to see when the inoculum attach the medium, yeah, or your trap, it will be like this picture. Yeah. This is Basidiospore of Exobasidium vexans that infect T3. So you can see there is some Basidiospore that we can catch in the spore trap. This is the other picture. You can see there is some cladosporium or the other pathogen that we can monitor from this uh, air trap, yeah. And it's only effective for spore, yeah, for fungi spore. It still cannot to monitor bacteria, virus, or nematode that probably move in the air. Okay, guys, so that's all our material for this week. I hope you get a lot of knowledge or information for today, and you can use it for your research or for your situation track. Thank you. If you have any question, please send to the WhatsApp directly to me or by using groups. Yeah. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.